Good afternoon, everybody. Welcome to the latest edition of the REPROCELL webinar series. Today, we're going to hear about two different methods for using PCR to study iPSCs. The first presentation will be from Dr. Tsukasa Shugo at Genohead Bio, who's going to talk about their technology for using PCR to improve genome editing, CRISPR genome editing. And in the second presentation, Dr. Liam Brennan from Systemic is going to talk about their technology for analyzing the stem cell, the presence of stem cells in cell preparations. Thank you very much. If you have any questions, please use the Q&A button at the bottom of the window to submit your questions, and we'll address them at the end. Thank you for your attendance. Dr. Shugo?
Yes. Thank you for giving me a nice opportunity, Bob. I'm Tsukasa Shugo from Genohead Bio. Genohead Bio is a Takeda spin-out company. So it's a pleasure to have an opportunity to share some of our work on the CRISPR genome editing today. Okay, so I'm going to be focusing really on genome editing services. But I think before I move on to that, I just want to very briefly touch upon CRISPR Sniper, where we started a company on this technology platform, improving the knockout, knock-in, and SNP conversion. So the relationship to the digital PCR. Nowadays, many disease-related mutations have been reported. As you can see in this round graph here, about 60% of the causal mutation is point mutation. Even though it does not cause some diseases, the sensitivity of the medicine could be different by SNPs. On the top right, this is actual survival curve in cancer patients with treatment of anti-cancer drug. And obviously, we can see a clear difference between a patient with GG type and a GC type. Considering such knowledge, it is generally accepted that we need to think about genetic variation in clinical studies, which is now called patient classification. But patients have a different genetic background other than the focusing SNPs. I mean, genes, many genes could be different, like this. The cost of the clinical study is very expensive as well. So we would suggest a very simple approach, simply changing the SNP using genome editing technologies, especially on iPS cells. Such cells are really powerful because, iPS cells can differentiate into any cell types. So we can get various disease models with same background in any cell types. So we think this is a really essential technologies in drug development. I'll show you an example from publications, Nature methods, in terms of the knock-in percent of SNP conversion. On the left hand, the authors introduced 13 SNPs in iPS cells, and then tried to isolate the mutants. But the knock-in percent was in red circle, 0.04%. So they seeded 3,000 cells per well at first round. If weak signals were detected, second expansion from the wells were carried out to concentrate the mutants. As a result, the knock-in percent increased to the 0.73%, and finally, third round it reached to the 6.8%. This is not the special cases for SNP conversion. Knock-in is often low efficient. So how we can detect how can we detect such rare mutations? If you are thinking about qPCR, it's very challenging because the sensitivity is not enough to detect the 0.04%. In case of digital PCR, a PCR reaction mixture is distributed into the 20,000 wells on a chip like this, during which genomic DNA molecules are also distributed in the wells with a concentration of 1 molecule per well or less. Okay, 1 molecule per well or less. So this is a kind of limiting dilution in terms of DNA molecules. So after the PCR reactions, by counting the positive wells, we are able to know how many mutation molecules in the epidermis tube we had at the beginning. So this is a principle of digital PCR or dPCR. Theoretical sensitivity can be calculated by 1 positive wells per 20,000 well equal 0.005% theoretically. But actual sensitivity would be 0.1% or less. But it is enough to detect the low mutations by genome editing. Now moving on to the multicolored digital PCR, what we'll call sniper analysis. Sniper stands for the specification of newly integrated position and exclusion of random integration. It is very useful to detect the random integration, but here we would like to focus on the detection of rare knock-in percent. Actual procedure is like this. 7 days after transfection, genomic DNA is extracted for digital PCR for target gene and reference gene. Here is an example where we've done digital PCR with endogenous probe labeled with peak and exogenous probe labeled with FAM, respectively. If you are looking at the endogenous results, what you can see here, highlighted with pink, is a genomic DNA-derived signals because it's endogenous. If you are looking at the exogenous sequence probe, what we call sniper probe, FAM positive wells must have exogenous sequence, right? Next one is a green dot. Green dot is a double positive, peak and FAM positive. So double positive means exogenous sequence and endogenous sequence on the same molecule, DNA molecule. So knock-in genome. We can quantify the accurate knock-in percent with green dot numbers normalized with peak positive dot numbers. So this process is helpful to distinguish the interferences. You can see in blue dots like this. This is an interference resulted from the donor DNA or something. So this is a really good merit to distinguish the both by sniper analysis. Now we can analyze the knock-in percent in heterogeneous populations such as bulk culture. Bulk culture means just after transfection, which allows us to improve the genome editing scheme here. On the top, you can see a conventional CRISPR scheme here. After transfection of Cas9, the first thing you need to do is limiting dilution of the cell, not the DNA molecule of the cell. But you know it takes very long time from single cells to 100,000 cells. It takes 1 or 2 months, right? So if the isolated clones here don't have the sequence you want to make, we need to repeat this cycle from the beginning, which takes additional 1 to 2 months. This is a really waste. What's the problem? I think this decision point might be too late because after the hard works. So we put the decision point earlier, just after transfection and before the hard work limiting dilution of the cell with the help of SNP analysis. In the next slide, just talking about optimization of knock-in conditions. If you made no-go decision here, we can try retransfection changing the several parameters like this. For example, amount of guide RNAs. It's important because the guide RNA cutting efficiency is different by guide RNA to guide RNAs. And the transfection conditions, pulse 1, pulse 2, pulse 3 or something. And you can use the another guide RNAs. And importantly, amount of donor DNA is a really important parameters. And you can use knock-in enhancers. What's important is that this is not the optimization on model experiment conditions. This optimization is done with your own cells against your target genes. So this is why this system is very reproducible and reliable. And furthermore, this feedback cycle takes just 1 to 2 weeks. Very short cycle, we can improve the knock-in conditions. So now here is a go and no-go decision criteria from our track records. I think what you can appreciate it here is that we got a really nice knock-in percent in bulk culture. I mean, mostly more than 10% in bulk culture in the left side. Frequent question is, how much knock-in percent is enough to isolate the SNP mutants? Of course, theoretically, 10% knock-in gives us 10 mutants with 100 clone pickup, probably. Here is the actual data. If you are looking at the right side, we had eventually succeeded to isolate the homo knock-in clones and hetero knock-in clones. For example, in the last rank, 4 per 24 means 4 homo knock-in clones per 24 clone pickup, and 6 hetero knock-in clones per 24 clone pickup. So based on these data, we did see clear correlation between knock-in percent in bulk culture and isolation studies. Importantly, if bulk culture knock-in is really good, for example, more than 10%, even with less than 50 clone pickup are enough to get homo knock-in clones and hetero knock-in clones. So this is a really scheduled pickup. So scheduled pickup is a great merit of CRISPR sniper. If we imagine less than 1% knock-in, which is often seen in conventional CRISPR papers, I think that we need more candidate clones to find the mutant in isolation step. So 100 clone pickup or 200 clone pickup is really hard work. So we can save the laborious work by sniper. Okay, so the case studies. In case studies, we'll give you an example to prepare an Alzheimer's disease model using the CRISPR sniper. One of the hallmarks of the Alzheimer's disease is high A-beta 42/40 ratio, which often appears in presennin or PSEN molecule mutations such as P11L, proline 117 leucine. Here is the SNP site in PSEN molecules. Highlighted in blue is the SNP, C is wild, and T is a mutant. And we can design a guide RNA here. Here is a PAM motif. So it's very close to the SNP site. Very good guide RNA. What's interesting is that genome editing efficiency largely depended on donor DNA species. CRISPR sniper analysis are here, and we had some success in 300 nanogram of single-strand oligo DNA donor with knock-in percent of 4%. Here is a green dot, 4%. As the donor increased, knock-in percent was improved to the 8%. But obviously, we got better knock-in percent with longer donor here. Longer donor means 2 kilobase long, very long donor. So with the help of sniper analysis, we successfully identify good donor molecules and succeeded to pick up the heteromutant and homomutant from these conditions. Just showing the A-beta production after differentiation into the neuron from the iPS cells. Here is the A-beta 42 production, A-beta 40 production, and the ratio of the A-beta 42/40 ratio. So I think what you can appreciate here is that we got nice correlation between genotype and clinical pathology here. This is wild type. You can see the only C here. This is a homomutant. You can see only T here. Middle is a heteromutant. You can see the double peak here. Okay, so you might imagine as shown in this figure here, you can also integrate the GFP as an exogenous gene into the genome. If you target the differentiation marker as a DFP acceptor, such cells will behave differentiation reporter cells like this. I'll give you an example of cardiomyocyte differentiation in the below right, where red fluorescence proteins are fused in the differentiation marker, and the GFP are fused on a maturation marker. Obviously, after differentiation, you can see red fluorescence here, and after maturation, you can see GFP signaled here. This is one of the most popular applications of this knock-in experiment by genome editing. These cell lines are really useful for differentiation study. For example, it's useful to optimize the differentiation protocols. It's useful to screen the differentiation compound using a compound library. And also it's useful to detect the differentiated cells in organoid and so on. Here is an actual knock-in experiment where we've done GFP fusion on differentiation marker H. And before iPS clone pickup, we've checked the GFP knock-in in bulk culture using the SNP analysis here. You can see the green dot here. In other words, knock-in percentage is really good, 37%. So we tuned the pickup scale just for 24. I think what you can appreciate here is that we got a very high knock-in rate, 23 knock-in clones per 24 clone pickup. Furthermore, we also have the other examples showing very high knock-in rate, 16 clone knock-in per 16 clone pickup, 4 knock-in per 4 clone pickup. It's a really high success rate with CRISPR and SNP know-how. So finally, knockout is a relatively easy experiment, but hard to transfect cells are sometimes a problem. Often seen in primary cells, such problems. This is a knockout experiment where we've done CD4 specific guide RNA knockout on primary T cells, looking at the CD4 expressions by flow cytometry with bulk culture. And I think what you can see here is that we got a very nice knockout of the CD4 molecules, about 95% knockout by flow cytometry. So just to summarize the section, because Genet Bio is a spin-out company from Takeda Pharmaceutical Company, our services are staffed by professional team who are trained in Takeda, very high success rate. Knock-in conditions are optimized with your own cells, with your actual target genes. So this is why our system is highly reliable. And these services will give you nice opportunities in internal and collaborative researches. Finally, if you are not so familiar with genome editing, it's no problem. You don't have to do anything. We can serve anything, everything. Business flow. We have divided our services in 3 parts. First one is the design and feasibility study, in which we will prepare the genome editing tools, checking the cutting efficiency of the guide RNA, perform the transfection with 6 conditions. If you satisfy the knock-in percent in 6 conditions, and also you want to have the mutant as a clones, mutant isolations, the second step is scheduled cloning. And finally, third step is QC and making a stock of the mutant. So if you are interested in our services, please let us know by email or something. Then we will make the study design to have the discussion with you. The email address are here. Thank you for your attention.
Thank you very much, Dr. Shugo. Now we'll transition over to Dr. Brennan from Systemic.
Yeah, thanks for the introduction, Bob. Thanks to Casa, really good talk. I'm just gonna get remote control. There we go. Cool. So yeah, that's cool. Thank you very much. So Hi everyone, my name is Liam Brennan. I work at Systemic, and following on from what Tsukasa has spoken to you about today, I'm going to follow on with what we use digital PCR in the lab for. So while he was talking about applications for generation of cells, I'm looking at it in a way for screening cells for residual PSCs and a differentiated cell product. So, hang on. There we go. So Systemic we're a lab based in Glasgow in the United Kingdom and primarily operate as a service and product development company. We're mostly interested in using microRNAs as tools to support stem cell research and cell therapy product development. And our primary business is involved in using microRNAs in pharmaceuticals, biotechnology, and cell and gene therapy research for clients. So, as you're probably all aware, cell-based therapies show a lot of potential in a lot of different medical conditions. And in particular, in regards to stem cell therapies, the use of pluripotent stem cells are a more popular way of trying to produce novel derived cell therapy treatments. One of the risks of using a derived cell therapy treatment, however, is you always run the risk of having residual pluripotent stem cells remaining in the final derived cell therapy product. And of course, the main safety concern with this is after administering it to a patient, these residual PSCs can go off and form tumors, which is obviously quite a significant concern. So as a consequence of this, most regulatory bodies in most countries will have a requirement that a manufacturer of a derived cell therapy product needs to be able to quantitatively assess the level of PSCs in their products before they can release them for a clinical application. And even if a company is able to accurately quantitate the number of residual PSCs they have in their final product, a large number of regulatory bodies will make a request that they have a second independent testing agent to validate this to the same level or fewer residual PSCs than they were able to identify. And one of the things that makes this difficult is there is no real simple, established, standardized way to assess levels of residual PSCs in derived cell preparations. And it's for a number of reasons, but typically because if you're looking at quantifying PSCs in a prepared cell product, you need to have a very low number of residual PSCs before they'll pass any safety regulatory checks. So with a lot of the existing tests, one of the main issues is that they lack the sensitivity required. Or if they are sensitive, they're usually very expensive and very laborious in vitro or in vivo assays. Coupled with this, obviously, if you're preparing a cell product that you want to release for a clinical application, there's a need for the batch to be rapidly tested and the results given back to the manufacturer for quick turnaround of their cell product. So ideally, we're looking for something that you can use for routine quality control, but is also relatively inexpensive and has high sensitivity. So at Systemic, we developed an assay called System PSC Check. And System PSC Check uses Droplet Digital PCR to screen microRNAs that should only be found in residual PSCs in a derived cell preparation. And what we do for this is we have specific microRNAs and an internal microRNA control that we screen for in a cell product that can allow us to identify any residual PSCs in a prepared cell product. And I'll go into explaining this a little bit more later on, but one of the good things about the assay is that it's able to identify the absolute copy number of PSC-specific microRNA targets and how this will correlate with the numbers of PSCs in the preparation. So using this particular relationship, the assay is able to quantify the number of residual undifferentiated cells, and the PSC source can either be from embryonic stem cells or they can be from induced pluripotent stem cells, and the derived cell source can be any differentiated cell type. And this assay was developed and optimized in conjunction with the United Kingdom National Measurement Laboratory. Now, where we would offer this as a service is typically, like I said before, we have a client or customer that's looking to validate either a batch or they're trying to validate their own internal method. And we will receive frozen cell pellets of the cell batches from the client, and we can turn around a cell batch between 7 to 10 business days, depending on the number of cells. So it's fairly robust, but also very quick turnaround time for trying to get into those clinical applications. So to start off, why would we use microRNAs? So despite what most people think that microRNAs in humans are kind of useless, they are actually very important in regulating messenger RNA function. And that all the human microRNAs that are present are able to regulate about 60% of genes in humans. And this is things like diverse cellular pathways in both normal and disease situations. But in this particular aspect, what we're interested in is microRNAs have been shown to be very important in the maintenance of stem cell pluripotency and stem cell differentiation and self-renewal in these cells. From a biomarker diagnostic standpoint, what makes microRNAs useful to use is that they're very stable in pretty much every single form. So it doesn't matter if we receive frozen cells or we receive cellular lysate. The microRNAs don't degrade pretty much at all compared to a lot of other RNA or DNA that we've looked at in the past. And microRNAs are quite stable. You can get them out of tissues that have previously been put in wax that you've extracted out of that, which you can't really do for either DNA or RNA. So the reason why we look at digital PCR, Tsukasa touched on this a little bit before, but the main reason is digital PCR is a much more precise methodology to use than traditional PCR or qPCR. So here we have what a well would look like with a traditional PCR, and typically, in the most part, you only get a single fluorescent measurement or maybe 5 or 6 fluorescent measurements if you're trying to multiplex it. Now, the thing with digital PCR is that it technically carries out a single reaction within the sample. However, the DNA fragments are separated into a large number of partitions. And then in each individual partition, the reaction is carried out individually. So we can see here all these different droplets inside the well. So because we have this separation in this partitioning, digital PCR provides a much more sensitive measurement of nucleic acid amounts, and therefore is useful when you're trying to determine a very low individual number of targets in a sample. So what digital droplet PCR is an extension of this digital PCR method. And with ddPCR, what we do is we partition the samples in 20,000 droplets of a defined volume. So each of the target and background RNA or DNA get randomly distributed amongst the droplets. And then what happens is the PCR is amplified, and then the instrument is capable of reading the fluorescence in each individual droplet. And effectively what it does is it will measure whether or not each individual droplet does or does not contain the nucleic acid of interest. And then it will allow the absolute quantification of the molecules in the reaction. So the advantages of using Droplet Digital PCR is, as I mentioned before, performing the absolute quantification. And the way it does this is the software uses Poisson distribution in order to correct for multiple target molecules per droplet. So this provides some advantages because one doesn't need to worry about normalizing the target or having the ideal calibrated gene for the sample. You don't need to worry about running a standard in order to carry out absolute quantitation. Digital PCR has increased precision, robustness, and sensitivity in detecting low copy numbers. So if you're trying to find as low as 10 targets, individual cells in a population, it's very useful. And compared to other PCR types, it's relatively insensitive to any PCR inhibition. So in the development of our assay, what we did was we used a microRNA database to search for biomarkers that we think would be found in residual PSCs, so in iPSCs and undifferentiated cells. So to do this, we carried out a number of microarrays and qPCR reactions in order to find microRNA targets that were expressed at high levels in these cells. And in addition to that, they were not detected in high levels in derived cell therapy types by both microarray and qPCR. So from this original candidate of biomarker panels, we went and picked 4 which had very high expression in iPSCs and had almost no to very low expression in terminally differentiated cell types, which we've just labeled targets 1 through 4. So the way that we develop the assay is that we use samples with different levels of iPSC input numbers and a background of 1 million bone marrow derived MSCs. So this is achieved by either spiking a known number of iPSCs into a background of 1 million MSCs, or alternatively by spiking the equivalent concentration of iPSC RNA into a background of BMMSC RNA. So the first thing I want to show you is how the assay is specific for pluripotent stem cells. So in this graph here, we have three different groups. In the red, we have iPSCs. In the blue, we have embryonic stem cells. And in the green, we have non-pluripotent cells and terminally differentiated cells. And you can see for each of the targets that while we get high expression of them in both iPSC groups and ESC groups, the non-pluripotent cells and the differentiated cells do not have any expression. So this shows that we can use these 4 targets in order to screen for iPSC cells and ESCs, and we don't run the risk of detecting anything in differentiated cells. So in order to check the performance of the assay, we carried out a number of tests based on the ICH Q2 guidelines. And so we have a look at the target assay specificity, assay linearity, the detection sensitivity, and then we assess both the intra and the inter-assay performance, as well as having it independently validated by a different laboratory. And then we can go ahead and calculate the sensitivity and the specificity. So the first thing is that the assays themselves have greater than 99% specificity for their unique microRNA target, which you can see in the table here. So none of the assays have any cross-reactivity with any of the other targets. So you can be sure that if you're getting a positive for them all, it's not a case of one particular assay is actually giving you a false positive for one of the other targets. So there's very low to no cross-reactivity between each of the assays. In addition to this, the assay shows excellent linearity between targets 1 through 4 in respect to copy number and iPSC number. So in this experiment, what we did was we RNA spiked the equivalent of 1,000, 100, 10, or 1 iPSC in a background of 1 million bone marrow derived MSCs. And then, as you can see here on the graph, as the concentration of iPSC RNA increases, we also get increased expression. And this is the same. This is true for all 4 of the targets that we've screened for. Then, if we test the assay sensitivity with cell spike samples for all 4 targets, we can show that the assay is highly sensitive, and we can constantly detect both 10 and 5 iPSCs in amongst the background of 1 million bone marrow derived MSCs when compared to the control of just the 1 million BM MSCs alone. When we try it with the one single iPSC, you can see that it's difficult to distinguish it from the background. So while the limit of detection is somewhere between 1 and 2 iPSCs, we've set the LOQ of the assay between 3 and 5 iPSCs and a background of 1 million BM MSCs. So then we conduct the intra-assay analysis. So we have 2 different analysts conduct the assay using the same samples but different equipment in the same lab on the same day. And you can see that the assay has good reproducibility between the analysts, and it's not dependent on one person to get it to work. And again, you can see that we get good detection of both 5 and 10 iPSCs. But again, as we've shown before, the single iPSC is very difficult to ascertain against the background. Then if we get the same analyst to use the same samples on consecutive days, you can see that again, we get relatively consistent expression levels. So it is reproducible over different days as well. And then finally, we had the National Measurement Laboratory independently validated. And again, we have similar results as we see before, where we get comparable expression between the analysts for all 4 of the different targets. So then finally, what we do is we calculate the diagnostic accuracy of the assay, and we get no false negatives, so we can be sure that the assay has 100% sensitivity at both the 10 and 5 iPSC and 1 million bone marrow derived MSCs, and we never get any false positives either, so it has 100% specificity. So it's a very accurate measurement for trying to find very low copies of residual PSCs in a derived cell product. So just to quickly summarize, the assay has very good target specificity for the cells and is specific for pluripotent stem cells. The assays themselves have 99% specificity for the unique microRNA target and shows very good linearity between expression and the amount of RNA or the iPSC number. And regardless of day or operator or lab, the assay consistently detects both 10 and 5 iPSCs and a background of 1 million bone marrow derived MSCs. And the assay is highly reproducible, and we can get it validated by independent samples in different laboratories. So thank you very much for your attention today. If you'd like more information about the assay, then please visit our website or send an email to our general inquiry. Thank you.
Thank you very much, Liam and Tsukasa.